Hello and welcome to Sam Biology class. Friends, today we shall discuss about oxidative phosphorylation and chemiosmotic theory. Most of the free energy released during the oxidation of glucose to carbon dioxide is retained in the reduced coenzymes NADH and FADH2. During respiration, electrons are released from NADH and FADH2 and are eventually transferred to molecular oxygen. The free energy released during the oxidation of a single NADH or FADH2 molecule is sufficient to drive the synthesis of several molecules of ATP. The mitochondrion maximizes this production of ATP by transferring electrons from NADH and FADH2 to molecular oxygen through a series of electron carriers. The step-by-step transfer of electrons causes the free energy to be released in small increments that are stored as the proton motive force. This transfer of electrons from NADH and FADH2 to molecular oxygen through a series of electron carriers releases free energy that drives the synthesis of ATP from ADP and inorganic phosphate. The series of electron carriers constitute the respiratory chain. In eukaryotes, the respiratory chain is located in the inner mitochondrial membrane, hence it is also called mitochondrial electron transport chain. Whereas in prokaryotes, the respiratory chain operates in the plasma membrane. The electron carriers of the respiratory chain are organized into 4 supramolecular complexes. Out of them, complex 1, also known as NADH ubiquinone oxidoreductase, transfer electrons from NADH to ubiquinone. Complex 2, also known as succinate ubiquinone oxidoreductase, transfer electrons from succinate to ubiquinone. Due to this electron transfer, ubiquinone is reduced to ubiquinol. From ubiquinol, electrons are further transferred to cytochrome C by complex 3. Complex 3 is also called ubiquinol cytochrome C oxidoreductase. In final step of the respiratory chain, it is the complex 4, also called cytochrome C oxidase, that complete the sequence by transferring electron from cytochrome C to molecular oxygen, as a result of which it is reduced to water. In addition, another enzyme called FO-F1 complex or ATP synthase is also found in the inner mitochondrial membrane. This enzyme, which is composed of several subunits, drives the synthesis of ATP by utilizing the proton motive force. FO-F1 complex is composed of 2 main components, FO and F1. FO component, which consists of several subunits, span the inner mitochondrial membrane, whereas the F1 component is water soluble and it protrudes into the mitochondrial matrix. In case of prokaryotes, all these components are located in the plasma membrane. So during the transfer of electron from NADH and FADH2 to molecular oxygen through this series of electron carriers, free energy is released that drives the synthesis of ATP in a process called oxidative phosphorylation. Unlike substrate level phosphorylation, in case of oxidative phosphorylation, The energy of oxidation drives the synthesis of ATP. One of the widely accepted theory that explains the mechanism of oxidative phosphorylation is chemiosmotic theory. This theory was proposed by British biochemist Peter Dennis Mitchell in 1961. According to chemiosmotic theory, the proton motive force, which is established across the inner mitochondrial membrane during electron transfer, drives the synthesis of ATP. Out of 4 respiratory complexes, complex 1, 3 and 4 function as proton pumps. For every pair of electrons transferred from NADH to molecular oxygen, 10 protons are pumped from matrix to the intermembrane space. Out of 10, 4 protons are pumped by complex 1 and complex 3 each, whereas complex 4 pumps only 2 protons. So in the respiratory chain, complex 1, 3 and 4 serve as proton pumps. This is the inner mitochondrial membrane. This side is the mitochondrial matrix and that side is the intermembrane space. Due to the outward transport of protons, concentration of H plus ions in the intermembrane space increase, whereas the concentration decrease in the matrix. This difference in the concentration of H plus ions between the intermembrane space and the matrix constitute the proton concentration gradient. In addition, there is electric potential because the matrix is more negatively charged as compared to the intermembrane space. Both of these components, such as proton concentration gradient and the electric potential together, constitute the proton motive force, which according to chemiosmotic theory is the main driving force for the synthesis of ATP. It is because their concentration is higher in the intermembrane space, so protons flow back to the matrix through the FO component of ATP synthase. FO components serve as the proton channel. Movement of protons from greater concentration to lesser concentration cause the FO components to rotate, that in turn drive the synthesis of ATP. This is thought to occur by binding change mechanism in which the conformation of F1 beta subunits is changed as the axis rotates from one which binds ATP tightly to another which releases ATP and binds ADP and inorganic phosphate so that one more molecule of ATP can be synthesized. So friends, this is all about oxidative phosphorylation and chemiosmotic theory. For more updates, subscribe my YouTube channel, Sam Biology Class.